Jai Shri Gurudev. In this class we will discuss about excretion. As we studied in the previous classes, that is the digestion, respiration and the circulation. At the end of the digestion process, there will be absorption of the metabolites take place. The breakdown of the molecule gives out various metabolites like glucose, amino acids, fatty acids and etc. And out of the respiration process, during the respiration process, oxygen is diffused into this bloodstream. That is, from the atmospheric air content, oxygen gets diffused during the process of respiration, and which is the one other thing. And the circulation is the transporting system for both the metabolites whichever comes out of the digestion process and the oxygen which is necessary for the breakdown of the energy molecules like the metabolites. And after this three process is over, that is the digestion which gives out metabolites, the respiration process, the main theme of the respiration is to give the fuel, that is the oxygen, and for the transport of these metabolites and the oxygen to respective target cells or the terminal cells where actually the breakdown of the molecule take place. After the breakdown, that is after the cellular respiration is over, there will be a formation of various waste products like carbon dioxide. Once again, this carbon dioxide involved in the respiration process which will be eliminated out of the expiration. Meanwhile, various metabolites which are in excess quantity, which are not required for the normal leading of the life of an individual, and some other byproducts which are lethal or which are dangerous or toxic to the organism has to be eliminated. The metabolites or the energy molecules or any other molecule which are in excess more than the required quantity, which has to be eliminated out of the body. Meantime, the toxic material has to be eliminated out of the body. For this, there is another physiological process happening in our system, which is the excretion. In the higher vertebrates, which is the excretion process taken care by the kidney tubules. And various excretory products like ammonia product, nitrogen as such will not exist in our system, which combine with the other chemicals and form various nitrogen products like ammonia, urea, uric acid, creatinine. Likewise, various products, byproducts of this nitrogen combination exist in our system which has to be eliminated. On the basis of type of these molecules which are excreted by the animals are classified into four major categories. Now we will discuss four phenomenon. The first one is ammonotalism. Some group of organisms, most of the aquatic organisms excrete nitrogenous waste in the form of ammonia. Ammonia is the excretory product found in most of the aquatic animals, and like the protozoans, annelidans, octopus, pisces and tadpoles of amphibian excrete nitrogen waste in the form of ammonia, and the phenomenon excretion of the nitrogen waste in the form of ammonia, the phenomenon is known as ammonotalism. And those individuals like protozoan, annelida, octopus, pisces and tadpoles of amphibian, these group of individuals which excrete ammonia as a excretory product are called ammonotelic animals. The first thing is ammonotalism, a group of individuals which excrete ammonia as an excretory product, and these group of individuals which show ammonotalism are called ammonotelic animals. The next group is uricotelic, or the phenomenon is ureotelism. In the terrestrial animals, majority of the terrestrial animal excrete the urea, and for the ammonia the excess quantity of water is required, and ammonia is soluble in water. Ammonia is soluble in water, and this particular ammonia is highly toxic. For the elimination, it needs more and more of water, and which is found in the aquatic system. There is no limitation for the water availability, hence the animals can survive with engulfing more amount of water and eliminate ammonia. Similar way, in the terrestrial form, the urea is the excretory product, and which is less toxic than the animal, and which is also soluble in water, and which can be concentrated for some extent, and thus urea is eliminated in various organisms like human, camel, shark, kangaroo, rat, etc. And this phenomenon, that is the elimination of the nitrogen at waste in the form of urea, is known as ureotelism, and those animals which show this phenomenon are called ureotelic animals. And some other terrestrial individuals excrete nitrogenous waste in the form of uric acid, and which is less soluble or insoluble in water. Insoluble in water, therefore it has to be eliminated out of the body in the form of pellets. Like in insects, lizards and snakes and the birds, there will be minimum quantity of water present, availability of water. Since the uric acid is insoluble in water, the uric acid is eliminated out of the body along with the fecal content in the form of thick paste or white colored paste, and that is uric acid. And the phenomenon is known as ureotelism. And those individuals which show this phenomenon are called ureotelic animals or ureotelic organisms. And in few of the individuals, nitrogenous excretion is in the form of guanine. Guanine is the nitrogen excretory product found, like in case of scorpion and the spider. The excretion product or excretory product is guanine, and which is, the phenomenon is known as guanotelism. And those individuals which exhibit this phenomenon is known as guanotelic animals. These are the 4 different groups of animals which is on the basis of type of nitrogenous products, they are categorized into aminotelic, ureotelic, uricotelic and guanotelic. Next we will discuss on ornithine cycle, how the urea is formed by the cyclic process of ornithine. Let us discuss the steps. In the very beginning step, and thus ornithine cycle is happening in the liver. This is happening in the liver cells. And in the liver, the ammonia combines with carbon dioxide. This chemical reaction is carried out by carbamyl phosphate synthetase, and with the utilization of ATP molecule, 2 molecules of ATP get used up, and in the presence of a suitable enzyme, synthetase enzyme, ammonia combines with carbon dioxide form carbamyl phosphate. This is the first step of the ornithine cycle. Then this carbamyl phosphate, which is having ammonia group or nitrogenous group, carbamyl phosphate is combined with the ornithine, the amino acid, which combine with the ornithine in the presence of trans-carbamylase enzyme. The presence of trans-carbamylase enzyme, which couple the carbamyl phosphate with the ornithine and form citrulline. And later, this citrulline coupled with the aspartic acid, the citrulline coupled up with the aspartic acid in the presence of enzyme arganosuccinic synthetase with the utilization of ATP molecule and form organosuccinate. Organosuccinate get formed out of the combination of citrulline and aspartate. Aspartate gives one amino group to the citrulline and then form organosuccinate. Later, organosuccinate get split up into two molecules. One is arginine and another molecule is fumaric acid or fumarate. In the presence of organosuccinic cleavage enzyme, an enzyme involved in this process which splits up organosuccinate into arginine and fumaric acid. The fate of the fumaric acid, this organosuccinate with the enzymatic activity, the cleavage enzymes which split up into arginine and fumarate. This organosuccinate split up into two molecules, one is arginine and another one is fumarate. The fate of the fumarate is next, which will convert it into malic acid, which enter into TCA cycle, which is happening in the mitochondria. And the arginine get split up into or hydrolyzed into two molecules, that is hydrolyzed into ornithine and molecule of urea. So arginine get hydrolyzed in the presence of hydrolyzing enzyme arginase and giving ornithine and urea, and this is the urea which has to be eliminated out of the body. And ornithine once again which combine with carbamyl phosphate and continues its cycle. Let me brief up the reactions of ornithine cycle. This ornithine cycle is happening in the liver. In the first step, the ammonia combined with carbon dioxide in the presence of carbamyl phosphate synthetase with the utilization of two ATP molecules form carbamyl phosphate. This carbamyl phosphate combined with ornithine, the amino acid, forms citrulline in the activity of trans-carbamylase enzyme. Later, organosuccinic synthetase enzyme couple the citrulline with the aspartate and form organosuccinate with the expenditure of energy, that is ATP molecule is utilized. And later, with the enzymatic activity, organosuccinate split up into two molecules, one is arginine, another one is fumarate. This arginine in some of the physiological process enter into the formation of or synthesis of protein, whereas the fumarate or fumaric acid get converted into malic acid and enter into TCA cycle. And arginine in the presence of hydrolyzing enzyme arginase which get split up into ornithine and urea. Urea is for the elimination, and once again ornithine involved in the cyclic process of the routine activity of ornithine cycle. And now let us move on to another topic, that is physiology of urine formation. How exactly the urine formation take place? This the principal organ which involved in the urine formation is the kidney in the higher vertebrates. Kidney, inside the kidney tubules or nephrons are present, and roughly we can study the structure of nephrons here, that is, which has a cup-like structure called Bowman's capsule. Inside the cup-like structure, there will be a looping or coiled capillaries, blood capillaries are present, which are called glomerulus, and which leads into a tubular structure called proximal convoluted tube, and which leads into U-shaped structure called loop of Henle, and further which leads to a distal convoluted tube, and all the distal convoluted tube of the nephrons finally join to a common collecting tubule, and all the collecting tubule combine in the form in the region of hilus and through the urethra, which convey the filtrate. And with this knowledge, we will discuss the physiology of urine formation. As we know, whatever we eat up, which get breakdown into fine metabolites, which get absorbed in the blood by the intestinal wall, they later into the blood. From the alveoli of the lung, the oxygen get diffused in the blood capillaries of the alveoli of the lung, and after the metabolic activities, various digestive byproducts also given out into the blood. Now, in the blood, there will be useful as well as waste products present, and it has to be eliminated. The waste product has to be eliminated and useful product has to be absorbed, and water content has to be reabsorbed for maintaining the homeostasis. So, the physiology of the urine formation can be studied with the three phases. First one is glomerular filtration, second one is selective reabsorption, and the final last phase is the tubular secretion. Let us discuss one by one. In the blood, that is, the afferent blood vessel enter into the Bowman's capsule and which coiled up into network and exit out as efferent blood vessel. In the blood vessel, the pressure is high, and therefore in the blood plasma there are products like water, majorly the water content, the plasma water, in which glucose, calcium, potassium, sodium, chloride ions and various other useful products are present in the blood plasma. And the blood capillaries comes and coils inside the Bowman's capsule, during which the diameter of the blood vessel decreases. Therefore, the blood pressure increases. Therefore, the products like this water, glucose, calcium, potassium, sodium and chloride ions enter into the tubular structure of the Bowman's, and the content which is present here in the Bowman's capsule's tubule in this region, which is similar to the content of the blood plasma, except the blood proteins, blood plasma proteins and blood cells. The content of the filtrate, which is similar to the content of the blood, except the plasma proteins and the blood cells like WBC, RBC. Except the that means what? From the blood stream, the water enters, other mineral nutrition enters, glucose, anything which enters except the protein, blood plasma protein and the WBC, RBC platelet cells, and all other content here which is present, which is called primary urine. Primary urine, and this is known as glomerular filtration. The first phase of the urine formation is glomerular filtration, during which the diffusion of the molecule take place from the blood plasma, that is from the blood capillaries into the Bowman's capsule. And the first formed urine with all these content is called primary urine. Later, this primary urine get conveyed into the proximal convoluted tube. This is conveyed into the proximal convoluted tube. Here actually the useful and necessary products of the primary urine has to be collected or reabsorbed, collected back. And about majorly the 85% of water from the primary urine, which is conveying now in the proximal convoluted tube, get reabsorbed. Likewise, the energy molecule which is essential for the life, which is the glucose, also reabsorbed. And mineral nutrition like potassium, calcium, bicarbonate ions, sodium are also get absorbed. And this is selective process. Out of all this content, the water majorly along with glucose, potassium, calcium, bicarbonate, sodium and chloride ions, these get selectively reabsorbed in the proximal convoluted tube. And in the endless and remaining amount of water get reabsorbed, and in the endless loop particularly the sodium and chloride reabsorption take place. And the primary urine which is formed is approximately 125 ml per minute from all the kidney tubules. From all the kidney tubules of the kidney, per minute there will be formation of 125 ml of primary urine formed, out of which after this selective reabsorption from the proximal convoluted tube and the loop of Henle, only 1% of the content moves to the distal convoluted tube. 1%, nearly 1%, for say 1 ml. Out of 125 ml, only 1 ml is conveyed to the distal convoluted tube. In the distal convoluted tube, the large proportion of sodium get reabsorbed. Large proportion, highest sodium ions get reabsorbed in the distal convoluted tube, and small quantity of chloride also get reabsorbed. In the first glomerular filtration process, primary urine is formed. About 125 ml of primary urine it formed in this process. That means what? Per day there will be 170 to 180 liters of urine get filtered. The fluid get filtered per day from the kidney, from all the tubules. Out of 125 ml of primary urine, only 1% get enter into the distal convoluted tube. Now all this primary urine, there are essential molecules present which are get reabsorbed by the system, and this phase is known as selective reabsorption. In the beginning, the first phase is known as glomerular filtration. First phase is glomerular filtration. In the second phase, selective reabsorption of various molecules take place. Now, the filtrate is here in the distal convoluted tube, and which is about to enter into the collecting tubule. And during this, the various molecules like hydrogen ion, ammonia, and excess of the potassium, which is not required for the system, which is in excess. The excess form, excess content of these or get secreted into the tubule, distal convoluted tube. All these things, along with the pigments, various plant pigments, whichever we intake through the food, the pigments and the administered drugs like in the pathological condition we may take drugs in the form of tablets or injection. All those which are not necessary or required after the usage or get secreted into the tubules. Now, and this phase is known as tubular reabsorption. Tubular reabsorption takes place. Now, the filtrate is with the unwanted content get enter into the collecting tubule. Likewise, from each of the nephron which collects the unwanted substance which are now in the collecting tubules together. The first phase, the overall filtration takes place. In the second phase, the essential molecules get reabsorbed. In the third phase, that is tubular reabsorption, unwanted substances are get secreted into the tubule, where here it is reabsorbed, it is secreted into the tubule, and all that content present in the collecting tubule. And in the collecting tubule, the excess amount of the water get reabsorbed once again. The water of the filtrate get reabsorbed for the maintenance of water balance. And after these reabsorption of water, and the concentrated form of the content is known as urea, which is the final phase, we can call it as. This is for the excretion. This is how in human beings and other vertebrates the urine formation take place. There will be filtration of the molecule take place. In that, selectively some of the molecules are reabsorbed, and later in the tubular secretion, unwanted drugs, pigments, hydrogen ion, potassium, ammonia get secreted into the tubule, and all the nephrons collectively join to the collecting tubules. In that, further there will be reabsorption of water take place, and finally the concentrated form of urine, which is now said to be for the excretion. This is the physiology of urine formation. Thank you.